Hello everyone. Today we're going to talk about how to deal with a messy sequence. You guys are soon going to receive your DNA sequences back, and you will receive a string of letters as you see here on the screen. Now you can see in various places there are many nucleotides that are listed. Like for example here we have G, 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 C, C, G, T, G, T, C, T, C. And that's great. But you might notice that elsewhere in this sequence we see strings of the letter N. There's a long string here at the beginning of the sequence, there's a couple more here, and there's a string here at the end, another string here. Also scattered throughout the sequence you will see various positions where there is an N present. Now N means null nucleotide. What that means is that the machine that did the sequencing of your DNA sample was unable to identify which base correctly belongs in that spot. Now, there's a couple of ways to deal with this. The first thing we could do is we could simply trim off the beginning and end sequences, and then cut and paste the remaining sequence and put it into BLAST for a BLAST search. And it would probably work fairly well, at least with this sequence, because we have a lot of continuous identified bases without any ends in there. And the more we have in sequence without any ends, the better our chance of finding a homology to a known sequence in that database. But sometimes we end up with a result that does not have a great power score. The score that you get when you do the BLAST search tells you the confidence of that match. And sometimes it's not as good as you'd like, or you could make it even better. So what we can do is we can try and not just trim out the ends, but we can try and figure out what they should be. And the way you do that is you look at the data that the machine produces while it does the actual sequencing. This is the waveform trace file created by the machine that does the sequencing. Now when it does DNA sequencing, what it basically does is it tags different bases with a different fluorophore, and it looks for fluorescence given off by those particular sequences where they exist. And so it can read different colors emitted by the different nucleotides, and then you can use that color emission to tell you what nucleotide is at that position. So we see here the beginning of our sequence where we had a lot of the ends from the beginning in our text file. It's all these bases right here. The machine at the beginning of its run was having a lot of waves that were complementary to each other, overlapping each other, and it really couldn't determine what was what based on that. But then when it got to here, it started getting more confidence in what it was seeing. And by the time we get to here, we can see very clear peaks corresponding to different bases. So we'll take a closer look at some of these. Like for example here, right here. We look at this sequence here. We have this black peak, which corresponds to guanine. Then we have this blue peak, which corresponds to cytosine. Here we have red and red. That's thymine and thymine. So the machine is capably and easily reading the bases at this point. But every once in a while, it'll fall into a place where it just can't figure out what that particular base is based on the waveform reading. And we'll look at that here. Right here. At position 271, 2, 3, 4, 5, 6, it couldn't determine what this base is. And so it put an N in your text file for your sequence. But if we go back to this data and look at it, and we look more closely at what's going on here, we can see that there's a little green peak right here. Now, it may be masking another peak, it may be masking the blue, and maybe we can't see it, but it looks a lot like what's going on here and here. And so what I would do, if I'm trying to clean up my sequence, is I would look at this peak, and I would call that adenine, and I would insert A into this position in my text file. Then I would go on and look for other places like right here. Let's see if it's possible to look at anything there. Here we have this null nucleotide. And we look at this trace form file and it's getting really hard to see anything. The fluorescent peaks are very small. The machine's still able to read them, but the intensity is quite low. And at this point, even if we zoom in ourselves, it's very hard for us to see what's actually happening at that particular position right here. Now, I can see here a little bit of red going up and down corresponding to thymine. I can see a black right there corresponding to guanine. But right here, it's looking really flat to me. And so at this position, I probably would leave the N. I just can't tell what's going on there. So basically, that's what you would do, is you'd go through and clean up the single nucleotide missing results to see if you can figure it out. After you do that, you could try running your blast search again and see if you get a better result. Another thing you can try to do, if possible, is to work with this first sequence right here, because we have a lot of null nucleotides here at the beginning. We have signals, but we don't really know what to make of those signals. So if we zoom in a little bit on that, maybe we can kind of tease some things out. Like for example right here, position 30. I'll move that to the middle. Here at position 30, the machine is uncertain what it should call the nucleotide at this position. And that's because it has 2 peaks at that position. It doesn't really know what to do. It doesn't know if it should call it thymine or if it should call it cytosine. Now in this case, this is where you kind of make a judgment call. We do have a peak and a peak. They're pretty clear. But the intensity at this position is greater for cytosine. So I might go ahead and plug in cytosine at that position. And then I start moving backwards towards the beginning of the sequence and see if I can do that for any other position. Here, I've got relatively low signal for thymine, low signal for guanine, low signal for adenine, but I've got a relatively high signal for cytosine. So I would probably plug in a C at that position. I move back here and keep looking. Now here it's hard to tell. We have this big black trace of guanine, and it peaked out right here. But then it went down and didn't really peak back up again. Even though it's the strongest signal, it appears to be on the downslope. And so I wouldn't have real confidence that that's correct. What I might do then instead is look for somewhere where I see a signal increase at that position. And I see a signal increase right here. So I might go ahead and plug in cytosine in that position. Even though the guanine has a higher intensity, it's clearly on the downslope from its previous marker right here. Now we go to this position. If I look at this, I see a pretty good peak for guanine. I'd probably put guanine right there. So when you go through here, you would look at these and try to make those kinds of judgment calls. This position, really hard to say. We have multiple overlaps, different colors. We have guanine being the strongest, but it's on the downslope. But I do see something on the upslope here. That's thymine. Now because this is on the upslope, this is definitely on the downslope, and these are flat, I would give this a little more weight, and I might call this position thymine. Here, this definitely wouldn't be thymine. We have pretty flat signals here, one on the downslope, one flat, but we have what looks like a peak. So I would probably call this position guanine. Same for, that clearly identified this one here based on its criteria, but then we get to like this position right here. We have thymine on the downslope, a flat adenine, a little bit of a bump for cytosine, and clearly a dip, a reduction of signal coming down for guanine. So this one, I might plug in cytosine in that spot, the C. So as you get closer and closer to the front of the trace, it becomes harder and harder to tell these things apart. But you can look at these and try to use a little bit of deductive reasoning. Like for example here at position 10, I've got almost no signal for thymine. I've got a flat cytosine signal with a little bit of a dip. There is an upslope here on the adenine signal, and I've got a flat signal on guanine. Even though it's matching the adenine signal, that's flat, but this is on the upslope. So I would probably call this A for adenine. So you can go through, plug in these values into your trace as best you can figure out, and see if that improves your blast score.